Hi guys, welcome to Memo's Tutorials. Today we are looking at conditions that are necessary for germination to take place. That's very important. Firstly, we need water. This is necessary for chemical reactions to take place. It also acts as a medium for dissolving food substances so that the enzymes can actually digest the food easily during the respiration period where your seed must be supplied with energy, right? And you also need suitable temperature. We need a suitable temperature for germination to take place. That's why mostly you see in winter a few seeds can germinate in winter, right? Because it's way too cold. There is no suitable temperature there. But summertime, we've got temperatures about 20 degrees, 40 degrees going up like that. So the higher the temperature, it means the enzyme activity is also higher. All biological reactions that are taking place, they are also taking place at a higher rate inside your embryo. So the lower the temperatures, it means there's low enzyme activity. But when we are having high temperatures, not too high, just high temperatures, it means that your plant will be able to germinate easily. But take note, if the temperatures are too high, then you will have a problem. You will see your crops dying. They're just trying to germinate and then they die immediately because the temperatures are way too high. Okay, so what that means is that the enzymes have been denatured. They get killed if your temperatures are too high. And then we need oxygen. This oxygen is used in aerobic respiration. Oxygen is needed for aerobic respiration. So for seeds to germinate, they need oxygen, right? So this aerobic respiration provides energy for the plant, for the seeds to actually develop into a new plant, right? So now let's have a look at the experiments on conditions necessary for germination. Okay, so we are having our setup A, B, C, D, and our A, the temperature is 20 degrees. But then we're having a damp cotton wool, meaning there is moisture there. And then for B, when we have our seeds there, but then for B, the cotton wool is dry, meaning there's no water there, there's no moisture. For C, there's an oxygen absorber. An oxygen absorber simply means that there's no oxygen in C, though the seeds are there and the cotton wool is damp. But then at D, I'm sure you can see the temperature is 4 degrees. Everything else is there, but the temperature is too small, right? But with that being said, I'm sure you guys have got the idea already of what is going to be the outcome. Let's start in B. So in B, there's no water, resulting in no germination. No water, there's no germination. There's no way the food is going to dissolve. There's no way enzymes are going to be able to act on food because there's no water. So moisture was absorbed by the calcium hydroxide granules. That's what they use to absorb all the moisture. So no water, no germination. In C, they used alkaline pyrogalul to absorb oxygen. So once there's no oxygen, no germination will take place. No oxygen, no germination. And then in A, seeds will germinate. Why? Because all the conditions that are needed, they are available. Let me take you back to A. There is our A. The temperature is 20 degrees. There's a cotton dimple. There is oxygen entering there. So germination will take place easily. What about D? There's no germination in D. Why, guys? It's because the temperature was not favorable. 4 degrees, it's very cold. Because of that, there was no success in germination, right? So with that being said, we then move on to germination success. So this actually refers to how well a sample of seeds germinates when all the necessary conditions are present. When a farmer plants, let's say, throws down about 100 seeds, how many are going to germinate? All conditions are necessary, everything is okay. How many are going to germinate? That's what we call germination success. We want to see which one succeeded. But then I want you to know that when you grow, when you plant them, even if all the conditions are necessary, some seeds, they fail to germinate, but then some will. But the main reason why other seeds fail to germinate is because of lack of necessary conditions for germination. You know, when the conditions are not suitable for germination of that particular seed, it will stay dormant as if it is dead. It will stay as if it is dead while it is alive. But once we put the proper conditions, it will germinate. Have you ever thought of beans that we buy even in shop, packed, sealed? You know, at some point we might actually think that this bean seed is not living. But just put it in water. You will see it. It will start germinating. Okay. So, germination success is calculated from the results of germination experiment and is presented as a percentage, right? So, the percentage of germination or the germination rate, you simply say number of seeds germinated over number of seeds that were planted. Then you multiply by 100, right? So, for example, here we are having chipo.